Hello and welcome to another lecture on membrane transport, where we will be concentrating on facilitated diffusion, which comes under passive transport. That is, molecules are transported from a region of higher concentration to a lower concentration. That is a downhill process, the same as with the facilitated diffusion, but this is helped by protein transporters, maybe protein carriers or protein channels. And in this lecture, we will be concentrating on protein carriers, those carriers which carry glucose and other hexoses, namely glucose transporter family of carriers, that is GLUT carriers. Now, facilitated diffusion depends on concentration gradient, so it is downhill process. And in diffusion, molecules which gets transported actually gets dissolved in the hydrophobic core of plasma membrane. So this is unlike what happens in facilitated diffusion. Facilitated diffusion, normally it carries larger molecules, polar molecules or charged molecules which do not get dissolved in the hydrophobic core of plasma membrane. So they have to be carried inside, either by carriers or or through channels. Now, channels we will discuss in another lecture. Here we will be concentrating on the carrier proteins, that is the GLUT proteins which transport glucose and other hexoses. Now, glucose is a large molecule. It's a polar molecule, so it cannot go through the plasma membrane by diffusion. So this hydrophobic core, this will not allow the polar molecule, namely, or large molecule, namely glucose, to get into the cytoplasm, into the cell. So what happens is that the glucose depends on GLUT transporters. So this is what happens. Now, glucose cannot go through the plasma membrane, but it can take the route of GLUT. Now, glucose transporters are a wide group or a wide variety of membrane proteins that facilitate the transfer of glucose across plasma membrane and down its concentration gradient by the process of passive facilitated diffusion. And this is an example for uniport transport mechanism also, in that only one type of molecule is transported across, and that too in one direction only. Now, GLUT are integral transmembrane proteins, as you can see, and contain 12 transmembrane-spanning helices, 12 transmembrane helices with both amino group and carboxyl group. These termini are exposed to the cytoplasmic side of the cell, of the plasma membrane. Now, a GLUT family of proteins are like isoenzymes or isozymes, in that they differ slightly in their amino acid composition and do the same function of transporting glucose, but having slightly different properties among them. And since glucose is a vital source of energy for all live organisms, these transporters are present in all phyla, and it is found almost in almost all of the mammalian cells also. Thus, GLUT, or generally the carrier-mediated facilitated diffusion, is referred to as ping-pong mechanism also. This is because the carrier can exist in two structural conformations. Now, here we can see that this is represented by a glucose molecule, and this is the carrier, the GLUT carrier, and this glucose moves and it attaches to a specific site on this GLUT carrier molecule, and attachment of this glucose molecule to this carrier actually induces a conformational change on this carrier molecule, and it changes conformation, and as a result the affinity of the carrier towards this glucose molecule loses, and the glucose molecule gets like this, it gets released inside the cytoplasm. And once glucose leaves the molecule, the GLUT carrier, what happens is that the carrier again changes the structure and revert back to the original structure. So this is what this is actually the mechanism of action of GLUT transporters. Now, this is the ping pong mechanism which transports glucose from outside the cell to the inside the cell using GLUT. Now, there are at least 14 GLUTs available, and this actually allows different tissues to meet their specific or varying needs. And here we will be concentrating on the most important 5 GLUTs or glucose transporters, namely glucose transporter 1, 2, 3, 4 and 5. Let us see glucose transporter 1 first. So they are located on different locations or concentrated on different locations like this, and they have different properties also, especially the Km value also differ. So that is very important, you will see. Now GLUT1 is present in all cells of the body, and it is responsible for the basal glucose uptake of the body. The Km value of glucose, this Km value for glucose of this GLUT1 is 1 millimole, which is much less than the normal blood glucose level of 5 millimole. Now here 5 millimole means in milligrams it comes around 80 to 120 milligrams. So this is expressed here in millimoles because as to correspond with the Km value also. So 5 millimoles is the normal blood glucose level, and here the Km value of 1 millimole for GLUT for glucose means that they have very high affinity for glucose, and hence glucose enters these cells very easily. So that is a need of the body also, need of these tissues also, so as to maintain the blood glucose value at around 5 millimoles in blood. Now we'll go to GLUT3 because I am taking this for discussion now, because this transporter is also having a value of a Km value of 1 millimoles, and it is having a high affinity for glucose. Now GLUT3 is present in very high numbers in brain and nerve cells, and it is having the same Km value of 1 millimole as GLUT1. As we know, the chief energy fuel of brain is glucose, and so the transporter should have very should have very high affinity for glucose. So this is why this GLUT3 is having a Km value, very low Km value and very high affinity for glucose. And now we go to see the GLUT second, and GLUT second is present in liver and beta cells of pancreas, concentrated here mainly, and Km value is 17 millimoles. GLUT2 is having a very high Km value here, that is 17 millimole, which means that they have a very low affinity for binding glucose, and this is rather understandable with the positioning of these transporters, because they are concentrated on liver and beta cells of the pancreas, and in these tissues, that is in liver and pancreas, glucose entry needed to be allowed only when the available glucose in blood is sufficiently high. So when blood glucose is high, that is more than normal level only, especially like after a rich carbohydrate meal, only these transporters will be induced or expressed. Here liver can now start to store glucose, while beta cells of pancreas will be induced for the production of insulin when blood glucose level of blood level of glucose is more than the upper normal level. So that is what happens with this kind of Km value. Hence the high Km value of GLUT is highly significant in these tissues. Now we go to see glucose transporter 4, which is present in heart, skeletal muscle and adipose tissue, and this is the lone GLUT which is insulin dependent. So all others are insulin independent. So GLUT4 is concentrated in skeletal muscle, adipose tissue, and has a moderate Km value of 5 millimoles. Hence it can be specially seen that the GLUT4 is expressed in high amounts in response to insulin, because this is insulin dependent. This further helps to bring down the level of glucose in blood to normal level after a carbohydrate meal, in addition to the glucose being uptaken by liver and pancreatic cells. So this is obvious that this the Km value of this particular glucose transporter help to maintain blood glucose level, and apart from the work that is being done by liver and pancreas. And we have got one more GLUT5 here. This is having a function of taking fructose or transporting fructose mainly from intestine. So here we can see that the presence of GLUT isoenzymes actually fine-tune the glucose metabolism or glucose metabolic orchestra of different tissues in the body in this manner.